Thank you very much for inviting me to speak tonight. I'm sorry I'm labelled as Sylvia Clement. That's not who I am. I'm Liz Holden, and I've been interested in fungi now for at least 40 years. And Waxcap Grassland fungi is one of the groups that I've spent quite a lot of time looking at. So tonight I would like to introduce you to this group of fungi, and in particular to the habitat that is their particular niche. So wax caps, you can see on your screen, there's 4 pictures here of brightly coloured fruiting structures, fruiting bodies of fungi. And these are all examples of the genus Hygrocybe. Well, it used to be Hygrocybe, it's been split up. And you'll probably be hugely relieved, those of you that aren't mycologists already, to know that the common name for this group is wax caps. And we're calling them all wax caps, never mind how divided the DNA analysis has shown them to be. So wax caps are a particular group of fungi that do very well in these grasslands. They're not the only group of fungi, there's a whole suite of them. And as we go through the talk tonight, I'll introduce you to what we call the CHEGD species, that's CHEGD, and that will all be brought up on the screen later, but it's the sort of thing to introduce early on, so it's not a complete shock to the system. And that incorporates all of the fungi that we know of at the moment that particularly need the requirements that Waxcap Grasslands have. So we'll be looking at what those species look like and how you can identify them. But before we get on to the sort of the pretty picture bit of it, I'd like to talk a little bit about what this habitat is and why it's so important, some of the threats that face it, and some of the difficulties that we have as mycologists, people who look at fungi, in actually interpreting these sites and deciding how important they are in terms of conservation. So let's have a look. So this is a typical waxcap grassland, and the key to it is the short sward. It's generally dominated by grass. It can also have a lot of moss and liverworts in it, and a good range of herbs, although not always. But the key is that it needs to be short. And the other really important character is that the soil has to be nutrient poor. So it doesn't actually matter whether your sward is kept short by grazing, and that can be sheep, very often sheep, but it can be cattle or horses, or on coastal grasslands, rabbits will keep the sward short. It can also be kept short by mowing, and this is a particularly waxcap rich site in Aberdeenshire. This is Haddow House, which is managed by the National Trust for Scotland, and you can see nice straight mowing lines here. Fortunately, what you can also see is that these gardeners have taken all the clippings off with them, and if you need a nutrient poor soil profile, then obviously not leaving the clippings to increase the nitrogen is very helpful. So they've been very good at Haddow and have taken on board their wax caps there. So yeah, they can be mown, that keeps it nice and short. It has to be said that with mown grasslands, the niches, the number of micro niches within the grassland is not as great. So although the waxcap diversity will be really good, some of the other species may not be there. But anyway, if you're living in a sort of lowland area that's very agriculturally improved, sometimes those old lawns and perhaps unexpected niches, little corners, very often in churchyards, can be particularly rich for wax caps. So even, you know, in lowland areas where a lot of the grassland's been improved already, you can find wax caps, and churchyards can be a very good place to start. I don't want to label this point, but obviously all of these habitats and the whole waxcap grassland habitat is a managed landscape. It is a man-made habitat. And we think that probably in nature, these fungi would exist as very rare fruiters in woodlands. And indeed, you will occasionally find them in woodland situations. What we think is happening is that in woodlands, they get outcompeted by the other fungi in the woods that are actually living on the tree roots. And some of you will know this and others won't, but more than 90% of our planet's plants and trees have a fungal partner which enables them to exchange nutrients one to the other. And obviously those fungi are missing if you don't have the big trees. And we think that the tree fungi, they're called mycorrhizal, but don't worry about that, that they probably outcompete things like waxcaps. And that's why waxcaps have been able to do so well in grassland situations like this, where they're not actually in competition with the tree species. And something else I would say, I don't want to labour the point, but since it is out there, the phrase rewilding is very popular these days, and in something, you know, in contrast perhaps to what we're going to be suggesting, which is micromanagement in some respects. I suspect that rewilding is perhaps a hugely simplified topic, and when people really start looking below ground, which is what we'll be doing today, or what I'll be encouraging you to think about today or this evening, when people do start really understanding what's going on in the soil, they might actually value the ability to be able to pull out soils that haven't been hugely altered since the last ice age. And we know soil is constantly moved around by worms and tree roots and all of that. But the wonderful thing about waxcaps is that they pull out sites where the soil profile, so that's the actual soil, hasn't been interfered with. It hasn't been ploughed in very recent years. It hasn't been reseeded, and it hasn't been changed chemically by the application of farmyard manures or artificial fertilizers. So it's got a very low nutrient level. Soil profile is still pretty much intact. And I strongly suspect this is an extremely biodiverse habitat below ground. And below ground is a place no one ever thinks. We stand on the soil and never think more about it. But there are a few people now who are beginning to look at what goes on in the soil, and it's astonishing. And that is the actual foundation for any other changes and increases in biodiversity. That's where it all starts. So I throw that in simply to say there's a heck of a lot we don't know yet. And let's be a little bit careful before we just plough straight on into rewilding without maybe thinking hard about it first. Anyway, what about this habitat? We'll go back to Waxcap Grasslands and not get too tied up with all of those unknowns. These pastures would have been permanent pastures in the past. In Europe, the United Kingdom still has the vast majority of these unimproved grasslands, and even so. In an international level, the UK is important because, you know, these habitats just don't exist in other countries. We in the UK, although we still have such a large percentage of it, we know we have lost probably up to 90% of it since the last war, the big World War II, Second World War. The percentage figures are given as all different in different books according to when people actually looked at their figures. So we know that this as a habitat is becoming increasingly rare. And the reasons for that? Well, the obvious one is agricultural improvement, arguably necessary to feed a growing population. And by agricultural improvements, we're looking and thinking about ploughing, reseeding, and the heavy application of fertilizers. So this goes back and links into what I was saying about soil profiles that remain relatively intact. They've not been turned upside down by regular ploughing, and they haven't been chemically changed by the application of fertilizers. And to be honest, so much ground has been changed by those particular activities. We have lost huge amounts of permanent pasture and hay meadow. Other changes in land use could be forestry, just a reduction in grazing to promote flowering plants. That may not always be a problem, but we'll come back to that later when we look at management. Change of land use for housing. I'm sure you can think of very many other ways in which these often neglected, if you like, corners or unvalued corners have actually been put to, in inverted commas, better use. What else have we got? So alongside this need for low nutrient status, yes, okay, the application of farmyard manures and artificial fertilizers will do that. But something that we like to think we don't get a lot of, especially in Scotland, is airborne pollution. But in fact, when you look at the deposition maps, which you can find online, the threshold is about 5, and most of Scotland's well above that. So we do have nitrogen deposition in Scotland, not as much as in other areas, and certainly not as much as in large parts of Europe. We know a lot of the nitrogen comes from car exhaust fumes, and we also know that intensive agriculture can actually cause a lot of nitrogen to be deposited. And I know this is probably more a case in North America, but in the downwind of these big agri farms, there's a plume of nitrogen, which is incredibly intense. You know, it's a very high level of nitrogen deposition. So it is something to be aware of and for us to keep an eye on, because obviously if nitrogen is coming in from the air, that is also going to affect the chemical profile of the soil. A big threat to our Waxcap grasslands is perhaps simply a lack of understanding and awareness, and obviously that's part of what we're doing here tonight. It's often, you know, you lose these corners, these grasslands, simply because people aren't aware. They just think they're, you know, a waste corner of grassland, and they'd be put to much better use, you know, if they could get a small grant and plant it with native broadleaves. And that's done with the very best of intentions by and large. But it does mean that we sometimes lose them, and if people had understood more about what they had and the value of that land, that they might have thought twice about actually changing the way it was managed. And it's led to a lack of site protection. So our sites of special scientific interest, our suite of SSSIs, if we look at lichens in Scotland, there are over 70 sites that have lichens notified in their citation. So over 70 SSSIs with lichens getting protection. If you look at fungal citations, there are 8 sites where in fungi are notified. So and only 2 of those are for grassland fungi, and they're both on sites where actually the main drive of the management on those sites is the regeneration of woodland. So immediately you can see a bit of a management conflict there, and that's quite an interesting situation that needs to be looked at and taken forward and discussed. Climate change, well, that's anybody's guess. We really don't know what impact that's going to have on our Waxcap grasslands. We'll just have to wait and see. Okay, so we're aware that as a habitat, it's been declining. And we know that the Waxcap suite of fungi are very good indicators for soil that's not been improved, and that this actually means they will be very diverse for fungi, but also for the other organisms that live within the soil. So it's, you know, they are extremely diverse, biodiverse as habitats. I think perhaps for most of you who aren't mycologists, just a little explanation about what it is that we're seeing that will explain, hopefully, that statement below, which says primarily these are subterranean organisms. They only are currently detectable when they produce fruit bodies, and little is known about how and where they function below ground. So if you think of your umbrella-shaped mushroom, that's growing apparently out of the soil or wood or whatever, what you're seeing is just the tip of an iceberg. If you like, it is the apple, and the rest of the organism is completely hidden in the soil or the wood or whatever else the fungus is growing in. So the only function of those fruiting structures, those little umbrella-shaped mushrooms and toadstools, is to produce spores, and that's how the organism reproduces sexually. That's the method that they use. They produce these microscopic spores by the 100,000. But the real growing, feeding part of the organism and that produces those fruiting structures is a network of microscopic filamentous cells, And the individual cells are called hyphae. And perhaps the one word I would like you to take away with you is the mycelium. That's the name we give to the network of those individual filamentous cells that forms the body of an individual, a genetically individual fungus. So in fact, the fruiting bodies are only there for a very short time, but the structure below ground, the mycelium, in many cases, not in all, but in many cases, is there 365 days of the year, and it's functioning 365 days of the year. It's just not producing fruiting bodies. So it's very difficult to actually identify your fungi from the microscopic mycelium. You can't even do it with a scanning electron microscope. But very recently, what we have had just beginning to come on stream is the use of molecular science, DNA profiles and so on. And that is just early days, but it hopefully will begin to help us understand more about how these organisms are functioning in the soil. But if I say to you that in some circumstances, probably mostly in woodlands, but I dare say to an extent in grassland situations, that a cubic centimetre of soil can contain in excess of 2 kilometres of these tiny filamentous cells, if you were to put them end to end. This is an astronomical amount of biomass in the soil. And obviously there are going to be a lot of other organisms living on that and with it, things that we don't understand yet. I mean, it is just fascinating. We can send a man to the moon, but we can't make a chanterelle fruit properly in culture. So the only fungi you get in the shops are what we call recycler fungi, which grow on wood. It fascinates me, and it's one of the things I love about fungi is how little we know. So that presents all sorts of problems for us, because if each mycelium produced a fruit body every year, even though it's short-lived, you would be able to produce a comprehensive list of what was growing on your site. But actually they don't. And we know that some mycelium will be quite happy feeding and growing, but not producing fruiting bodies for up to 24 years. So the problem we've got is, are our fungi, fungal fruit bodies, are they genuinely rare? Or are they representing fungi which, if we're seeing the fruiting bodies very rarely, what I'm saying is that a rare fungus, or is it a rare fruiting fungus? So is it only a fungus that fruits rarely? We don't know very much about how extensive these mycelia are. And with this group of fungi, we don't know very much about how they're getting their nutrients. Very interesting. All those question marks mean that whatever management recommendations we make and whatever we suggest as being a rare and interesting site, you have to bear in mind that there are a lot of if, buts and maybes. And, you know, future work could suggest big differences. So we have to be a bit careful and, you know, and try and keep everything as generic as possible. We know that if one of these fruit bodies appears, that there will be wax caps there. And there are some which do seem to indicate that the site is going to be particularly diverse. And if you went back over lots of different years, you would actually start to find quite a rich diversity of the waxcap species. But it does mean that our distribution data is inadequate. And that is really the reason why waxcap fungi and fungi in general are so poorly represented in our SSSI system. It's just that people have been wary of actually designating sites for them because it was always felt that we didn't know enough about them. To compound all of that, many of the grassland fungi are very difficult to identify in the field, and you need to take them home and work on a microscope. Also, I mentioned DNA, and certainly as people are looking at what we consider identified fruiting bodies and getting a DNA profile from good material, what we're finding is that some of these species are in fact not one species, but a group of species that look very similar, but actually are genetically rather different. And that has certainly made us we've had to rethink the number of species that we consider makes a site important, even given the limitations I've spoken about. So there's a lot of work to do still on that. And sometimes it's difficult. You can see there are differences genetically, but they're not always, or sometimes we've used the wrong macro sort of characters to actually delimit our idea of what a particular species is. And of course, there aren't many mycologists around. There are very few of us really who are prepared to sit down with a microscope. If you compare us with botanists or ornithologists, very few indeed. It has to be said that at the minute, fungi are having a bit of a revival and people are genuinely interested, which is an absolute joy for us. And 20 years ago, nobody would have even thought about the possibility of a grassland SSSI, a grassland Waxcap fungal SSSI, whereas now at least we've got two, and there's a lot more work to do. But yeah, we are working on the profile of fungi, and people are gradually beginning to understand how vital they are, how important they are, that they underpin all of our habitats. So, okay, so sometimes when you have got a difficult group of organisms, you look at a better known group of organisms to try and tell you whether you're going to have a diverse site. So it was suggested that maybe any site that was good for its flowers would actually be good for its fungi. And a gentleman looked at this in, I think it was Sweden, did some work on it, and it was interesting. I then got the opportunity to do a similar bit of work in the Cairngorms, and I looked at, prior to it, somebody had done a broad brushstroke botanical survey, and they'd looked at the grassland around the farm, what we call in-by grassland, and botanically they'd said it was botanically poor, botanically moderate, or botanically good. And I took 8 of each of those, and I went back in the autumn to see what was actually fruiting in terms of fungi. And it was very interesting. What we found was that the really good botanical sites, yep, they were good for the fungi too, by and large. The moderate ones, yeah, they were moderately interesting. What was interesting for me was that the botanically poor ones could sometimes be brilliant for their fungi, even though they were actually designated as botanically poor. And we think this is because fungi are, as I've said, essentially they're a subterranean organism. So you can graze or a field to within an inch of its life almost, and the fungi will still pop up. They might be slightly trampled if you've got a very big horse or something grazing the horse, but they'll still fruit and you can still find them. Whereas the flowering plants are much more difficult to spot. And that's interesting because it means that by just relying on the flowering plant surveys, you're actually missing important sites where the soil profile is still actually good enough to produce a diverse range of waxcaps. And I think that really kind of pinpoints the importance of using fungi for these unimproved grassland sites. Interesting. So this is quite a hefty slide, but we have looked, I've said, you know, it's not well represented in our SSSI system, and JNCC and the various government agencies have been looking at how we might use fungi to actually designate waxcap grassland sites as SSSIs. This was done in 2018, and it's available online. It's chapter 14 is on fungi. There are other groups of fungi in there as well. And this is where you'll find, I mentioned right at the start, CHEGD, and CHEGC stands for, well, Claveriaceae, don't worry, but it's the coral and the fairy club fungi. H stands for Hygrocybe, which is the wax caps. E is Entoloma, and these two are genera, particular genera, that's contains several genera. Entoloma, they're the pink gills, they have pink spores. Geoglossaceae are the, Geoglossaceae are the earth tongues, so that's CHEG, and then D is Dermaloma. Now, we're not going to worry too much about those. They're a little bit more difficult to work with, and they're not that many of them anyway. The one that you will be most interested in, as most people are the wax caps, because there are a lot of quite good textbooks on them and some fairly straightforward keys that you can access online for them. But you can see here almost a reflection here. You need 19 at least wax caps in your grassland for it to be considered worth a closer look. And in addition, you need to have high diversity indicators. Here you go. And that is actually based on expert opinion. And I suspect that that will vary regionally, and that some will be common in some areas of the country, fruiting commonly, and in other areas of the country, they won't be very common at all. So our high diversity indicator species may turn out to be quite regional. But it gives you an idea of the sorts of numbers. And it doesn't really matter if that's just in one visit or many visits. But if you're thinking about a site being really, really good, you need to be able to distinguish at least 19. And the suggestion is, because actually distinguishing 19 is quite difficult for the average person, that if you can get up to 12 and then get hold of somebody who is prepared to come and have a look with a microscope at what you've got, then your site is almost certainly worth having a closer look. So that's really where we are at the moment in terms of our efforts to try and use wax caps to pinpoint particularly diverse waxcap grasslands. But as I say, lots of if, buts and maybes, and these numbers will undoubtedly, these threshold numbers will undoubtedly change. Okay, so let's have a little bit of a breather from all of that. When you go into a field, you are going to find species that aren't members of that CHEGD suite, the Waxcap Grassland suite. You will find any number of species. If your grassland is managed by animals grazing, you'll find any number of species that particularly associate with dung. And these will be found anywhere where there's dung. They don't need improved soil profiles or low nutrients. They very often have very dark spores, dark brown spores. And if you take spore prints, you will fairly quickly realize when you're looking at a dung fungus. So agaricus, again, likes quite nitrogen-rich fields. So you will find them occasionally in waxcap grasslands, but in general, they suggest it's probably a bit nitrogen rich. Again, they have very dark spore colors. And if you've got any trees in hedges, and possibly sometimes you'll get single trees left in a field, you may well find fruit bodies of some of these fungi that are actually growing on the roots of those trees. So these happen to be russulas growing with an oak tree. And all of these really are nothing to do with the actual waxcap fungi themselves. But it's just to flag up that not everything you find is going to be a target species. So just to remind you what the targets actually are: C, Clavariaceae, the fairy clubs and corals; H, Hygrocybe, which is the wax caps; E, Entoloma, the pink gills; G, Geoglossaceae, the earth tongues; and D, Dermaloma, the crazed caps. So let's just have a look and see what they actually look like. Hurrah, a pretty picture. This is the apricot club. I've put the scientific name there as well. I certainly don't expect people to remember them. It's a lot of English new names for you if you're not familiar with them. But somehow when you start, the English names perhaps are more accessible. But for those of you that know, that's the genus name and that's the species name. So Clavelinopsis luteolauba. Let's see. Here's some more fairy clubs and corals. White spindles. Clavarius flavipes, which was previously known as Straminia, the straw club. This was thought to be a very rare fungus indeed, until we started looking in a bit more detail at Waxcap Grasslands, and suddenly there it was, popping up all over the place. And the spores of this are on little cells on the outside of this top part of the club. And then you can see the slightly darker bit underneath. That's the sterile bit, lifting the fertile bit up into the air currents. And here's rose spindles. Really is that wonderful cerise pink. It's a lovely thing to find. And if you do find this one fruiting, it tends to come up every year in the same place, which is rather obliging of it when you're wanting to impress people. But not a particularly common species, the rose spindles. Golden spindles. This is quite a chunky thing. It tends to grow in these lovely tight clusters. But it's not branching, and you'll see what I mean about branching in a minute. And when I find this particular club fungus, I get a little bit excited, as I do with this one, which is smoky spindles, Caveria fumosa. I think they're both indicate that the grassland is of some value and age. And this one, of course, the beautiful violet coral. And you can see here that they're not just single clubs. These are more coralloid, and basically that what they've done by branching like this is increase the surface area for the spores to be released from. Here's another one, not quite so beautiful, but still a nice one, the beige coral. And a much more common one, the meadow coral, which if you start looking in this habitat, I can almost guarantee you'll find it when you get to an old, unimproved pasture or lawn. The meadow coral will be there. Right, that was C of CHEGD, and this is H of CHEGD. And this is the group that I suspect, if any of you do decide to look at the Waxcap Grasslands in a bit more detail, this is the group that you'll most be wanting to look at. So there are fortunately some very good field characters that will help you with your identification. And it's always important to know what you've got to look at. So I always say to people, never go through an identification guide and just try and match what you've got to the photographs, because you're going to make really horrendous mistakes. You need to know what you're looking at, and then go to your book and read the text. And if you've got a key, of course, that means you can actually get there much more directly. So when you're in the field, I would always take a notebook. A lot of these things that you can actually take the mushroom home with you. And I should say at this stage 2 things about collecting wild fungi. One is that even if you collect a very toxic one by mistake, you can't absorb fungal toxins through your skin. So if they are safe to handle, just follow sensible guidelines. Just wash your hands as you always would when you've been out in the field before you eat or drink. And the other thing is that when you remove a fruiting body from the field, you're not damaging the organism. Remember, the bulk of the organism is in the soil. And as long as you just take what you need in this case, to help you identify it, you will not damage the organism itself, just in the same way that you don't damage an apple tree by picking an apple, or a bramble by taking the brambles. It's a little bit different, obviously, with the edible fungi in areas where, like the New Forest and the Forest of Dean, where they're very, very close to large centres of population and cafes and restaurants paying good money for edible wild fungi. Because as I said to you, you can't persuade some of these fungi to grow in a commercial situation. We don't yet fully understand what makes them fruit. So where was I going with all of this? Yeah, so you can actually collect your fungus, and that means you can have a closer look at it. And in the field, if you're looking to Waxcap, always smell it. Smells, not everyone will be able to smell them, and the smells that you'll see written down in the book are not, it says it smells of honey. Now some people go, oh yes, honey, and others will go, can't smell anything at all. Some of them smell of rubber or oil, and there's one that smells a bit garlicky. So write that can be ephemeral. So do write it down and do share it around and see if anyone else can smell it, even if you can't. There's one beautiful little, very small white waxcap that smells intensely of Russian leather or pencil shavings, which is probably cedarwood shavings is more familiar, but it's so intense. And yet some people just can't smell it. For me, I can smell it, you know, without even going down to them if there's enough of them on the floor. So yeah, smell can be important. There's one that's very, very bitter. So you can just taste it, not actually, don't have to swallow it. You can just put your, in fact, that one, if you just put your lips to it, you can tell that it's bitter without even nibbling it. The others you can pretty much do back home. The texture of the cap and stipe is important. Is it dry? Is it slimy? Is it slippery? You know, is it greasy? Or is it really glutinous? Is it fibrous? Is it smooth? Is it scaly? What are the gills doing where they attach to the stem? Do the gills run down the stem? Do they come across from the edge of the cap and then go down the stem? Or do they come from the edge of the cap and then go up towards the cap or somewhere in between? That can be important. What's happening at the gill edge? Is it the same color as the rest of the gill? The gills are the plates of tissue that hang down underneath the cap, and that's the fertile part of the fruiting body. That's where the spores are, on the gills. What was I going to say about gills? Gill edge, no, that's fine. Width of cap and stipe, yeah, that's just a size thing. That can be important. And then you've got to decide whether it's just an immature fungus that's going to get bigger, or whether it's actually as big as it's going to get. And that comes with experience. And sometimes people find it very difficult to know when a fungus is mature, when a fungus is over mature, because sometimes they look as though they're dead, and actually they're not, they're in perfect condition. But it's a question of experience, I think, and going out with people who do know these things can really help, you know, with all of this. Shape and color of the cap, that can be important. The color of the stipe can be different to the color of the cap. And sometimes the colors will change. If you cut the fruit body in half or bruise it, you'll see one that is a sort of a gray color that goes a beautiful crimson red when it's bruised. So let's have a look at some of these characters actually on fruit bodies themselves. Here we've got a lovely smooth cap with gills that run down the stem. Here they are coming from the edge of the cap, the bottom edge, just slightly running down the stem there, definitely not going back up towards the cap. And here you can see that the stipe is quite pale and the cap is just a nice brown color there. This one has an earthy smell. And here you do have to look at the gill attachment. I don't know if you can see it very well. It comes across and then it dinks up and then it comes down. So we would call that sinuate. And that's very distinctive for this particular one, the earthy waxcap, because it comes in a range of colors from sort of pale brown, as this one is, right through to a much darker gray brown. And sometimes it has scales on the stem, and some people think then it's a different species. Yep, so there's lots to think about. This one shows you gills running down the stem, very much so. Oh, sorry. And again, this is the same thing. This has a very dry, matte cap. And although this looks a bit pale in this image, they can be very bright orange, right through to almost white. And again, there's a sort of white form, which some people think are a different species. Here's a very slimy one. Now this is, as it says, the slimy wax cap. It's grey cap, grey stem. And actually when you go to collect this, it's a real job to keep it. You know, you go to pick it and it just slips straight out of your fingers. And I suspect this is probably to try and keep the fertile part, the gills, because the gills themselves are dry. So it's just the cap and the stem that are slimy. To keep it all hydrated before the spores are released as they're maturing, and maybe to try and stop a certain amount of invertebrate grazing of the spores, because they're very nutritious. are the spores. Well, they're the most nutritious part of the organism as far as invertebrates are concerned. This is another slimy one, the heath waxcap. And this often, as the name suggests, this often tends to grow in more acid grasslands. And you can see that the gills are running down the stem. Now, in this one, here we go. This is the other thing about gill edges. I don't know if you can see it, perhaps better on some of these at the side, but there's actually a layer of gluten which runs right the way along the edge of the gill. The very bottom edge of the gill has a layer of gluten on it. And you can see it there. So that can be important. And you can see here that the gills are running down the stem. And this is the parrot waxcap. It comes, it can be very bright green, and then it fades to quite bright yellow, and sometimes it has pink tones as well. And that's really why it's called the parrot, for all its wonderful colours. It's another very slimy one. And actually, although it looks a little bit as though they're running down where these are also tucked up, if you look at this one, you can see that the gills are heading back up towards the cap itself. So they don't really come down the stem. But that's generally not difficult because it'll have these lovely bright green colours somewhere on the fruiting structure. And green's an unusual colour in fungi in general because fungi don't have chlorophyll at all. They don't get their nutrients from the sun through photosynthesis. Right, this is the glistening waxcap. This is another one with a viscid gill edge, decurrent, and a layer of gluten on the stem, and lovely viscid cap and stem, but this time it's in beautiful orange, yellow. Oh, this is a smelly one, and this has a very dry cap, but this one smells strongly nitrous, very chemical smell to it. And this is quite a robust species. You can see there that compared with the last one, which has quite a slender stipe or stem, if you compare it with the grass, it's not a lot thicker than the grass there. And this one, you can see the grass, this is a much more robust waxcap. Well, this is the very dull looking grey brown waxcap, the blushing waxcap, and you can see where it's been bruised along the edge of the gills, and gradually wherever else it's been bruised, it goes this amazing crimson red colour. And people often overlook this, thinking it's something that's gone over, and it's a question of getting your eye in. But this one is a real special species, and if you find this on a waxcap grassland, I, for one, get excited about it. Oh, and bless her, the ballerina, the pink waxcap or the ballerina, another beautiful cerise pink waxcap with a dry, well, sometimes viscid, but usually dry, a smooth cap, and a white, generally a white stem, this lovely pink cap. And you can see that very often species will change shape as they develop. So here it's looking very conical. And if you didn't know that as it grows, it expands, you could be looking for something that's completely different and not realize at all. And that's why I say don't just go through the photographs, because your fungi never look like the pictures in the book. Well, very rarely in my experience anyway. Here's another one that changes color. This is the blackening waxcap, and it starts lovely orangey yellow colors, and as it ages or you touch it, it goes black. And this is one of the ones where DNA has shown us that there's not just one species, but actually 11 blackening waxcaps, hence SL, which means sensu latu. So this is the blackening waxcap in the broad sense understanding of the concept. And the problem is there aren't enough professional mycologists, in other words, mycologists who are actually employed in mycological taxonomy, to match the DNA findings to the macroscopic characters. And, you know, we need to be able to do that to properly distinguish them in the field. And you need keys, and they're just not written yet. So that's why I say there's a lot of species delimitation still to be done. This is the persistent waxcap, because actually this one does stay persistently rather conical, even as it's got older. It's one of our yellow ones, occasionally with an orange tinge. and it has a fibrous stipe. And here's, sorry, I've gone a bit quick, but that's fine. This is another yellow one with a beautiful smooth cap and stem. And you can see the gills here, they're not running down the stem at all. They've gone back up towards the cap there. And that's the golden wax cap. It's quite a few of them, and this is just a selection. And yes, you begin to see that, you know, to distinguish the different, you know, the differences between the same colors, even though they are so beautifully colored and so delightful to see, that actually narrowing down which ones are different does take a bit of practice. This is a small, delicate one, quite a slender one. This is the Fungal wax cap. And this one often, but not always, has a red collar, and that one doesn't, as it happens. The citrine wax cap, these amazing yellow-green colors. It's a beautiful fungus. And here again, the gills are going up towards the cap. The gills themselves very pale, and the stem and the cap are these lovely lemon colors. Oh, here's another smelly one. This is one that smells oily. Some people say it smells like bed bugs, but I've only ever, in all the years I've been doing forays, met one person who knows what a bed bug smells like. And that was a district nurse who said, yep, that smells like bed bugs. So if you find this fungus with its lovely sort of almost salmon colored gills, it's quite robust. It has a yellowy cap and it's got this weird oily smell. Now you know what bed bugs smell like. Oh, here's a cracker. This is the orange wax cap. Often with this like a white bloom on the stem. You can see it there and you can see it here, but not always. And you can see that the stem is dry and the cap is slimy and viscid. Beautiful thing. Again, the colors do vary from yellow right through to this incredibly intense orange color in this one. And this is another species that gets me quite excited when it's found on a site. This is relatively common. It's the scarlet wax cap with a lovely bright red cap. It's not usually a massive fungus, but it can be, what's that, up to a couple of inches across, which is what, 3 centimeters, 4 centimeters, something like that. But this lovely red color and very often a yellowish stipe, very characteristic of most grasslands, waxcap grassland sites. Another red one, but this one tends to be much chunkier. And actually, can we see on this one? Where's it gone? Is that it? Yeah. You can see the gills coming down very slightly. They don't come right down, but there's a tendency for the gills to come down there. Whereas on the splendid wax cap, they're disappearing back up towards the cap again. They're not running down the stem at all. And as it dries, this one smells of honey. It's one of my favorite wax caps. I think it's absolutely beautiful. Sometimes the wax caps don't have a smooth cap. You can see here, this is minutely scaly on the cap. This is a small one. And here it's got like a very dark, almost a patina on there. That's the shadowed wax cap. And this one is Helobia, is the garlic one. So that's the one that smells faintly of garlic if you've had it in a container for a while. And another one, the goblet wax cap. You can see beautiful here, all these tiny little scales on the cap itself that are integral to the cap and important in helping you to identify that it's different from everything else you've seen. This is a nice big chunky fungus with a smooth greasy cap and a dry fibrous stem. And here's another chunky one, orange, with a dry fibrous cap and a dry fibrous stem. So you begin to see there are combinations of characters, and it's all about combinations of characters with all of the fungi really, but it's certainly very important in wax caps. And I should say that if you take spore prints, the spore color would be white. And if anybody wants to know how to do that, we can talk about that at the end. Oh, and just to throw this one in, if by any chance you happen to find it, this is a priority species. This is the date wax cap. Looks a bit like a blackening wax cap, but it doesn't blacken. It's this lovely date brown on the cap and then yellow underneath the cap. The gills are yellow and the stem is yellow. It's a beautiful thing. I suspect it's quite a rare fruiter. No idea how rare it is below ground. But if we have this, sometimes when you have hot, dry summers and thin soils, this will pop up and then you won't see it again for years. And 2 summers ago, it was popping up quite regularly. I wouldn't say it's everywhere by a long chalk, but it is out there. And I think I suspect that maybe it does need a hot, dry summer for it to fruit once it starts to rain in the autumn. And that's it again. Okay, phew, that's a selection of wax caps with a selection of different field characters. Don't worry so much. The rest you can, we'll just flick through just to give you an idea of what they look like. Here's Entoloma, one of the Entolomas, a pink gill, doesn't have a common name yet. These would have pink spores, whereas your wax caps have white spores. And they come sometimes in beautiful blues. And sometimes the blue is actually on the very edge of the gill there, on the bottom edge of the gill. Blues, dark blue greys, almost black. Corvinum, the crow, pink gill likes base-rich soil. So sometimes actually knowing a bit about the geology can help, because whilst most of them will grow in most sites, there are 1 or 2 that either like it acid, like the heath waxcap, or quite base-rich, like this one, the crow one. And this one, which is lovely yellow-green colour, quite bright and unmistakable to look at, even before you smell it. And anybody who's ever kept mice or hamsters, and you've had to clean out that corner in the cage that is used, you will know the smell. And this is exactly what this fungus smells of, hence its name, the mouse pea pink gill. But it does like base-rich soils. So that's just a little flavour of the pink gills, the Entolomas. And then this is the G of CHEGD, and these are the earth tongues, Geoglossaceae. So again, just a selection. You really need a microscope for most of these, same as the Entolomas. It's very difficult to identify them to species without a microscope. They all tend to look very similar. So here we go. That one's actually a viscid earth tongue, so that is different if you get that. And sometimes they will grow in clumps, but not always. They're very often just singles like this. Sometimes the microglossum can be this lovely red-brown or a beautiful copper green. They're very lovely when you find them, very nice. And there is, I should warn you, a woodland version of this which has a scaly stipe called Microglossum viridi. This one has a smooth stipe. If you find it, if you find a green one in the wood, have a look at the stipe. If it's scaly, it won't be your grassland species. The grassland one has a very smooth, shiny stipe. And this is D. I did mention D, CHEGD. There is a small group of other genera. This one I've put in because it very often has this cracking cap, and that's to do with the structure. That this very top layer of the cap has round cells in it. They're not spaghetti cells when you look at them down the microscope, and it just means that as it grows, sometimes that the round cells pull apart, and the effect of that is that you get this crazed pattern on the top, like the crazing on ceramics when they've been through the dishwasher too many times. It also smells mealy, and mealy is best described as either rising bread dough or fresh milled flour, something like that. It's quite a distinct smell once you get used to it and get your nose into it. So I just show you that for completeness. Okay, just to try and pull some of that together, management of waxcap grasslands. The things that we try and suggest to people they do to maintain the grassland with its fungal community: keep the sward low, avoid it reverting to scrub or woodland, which will happen if you don't do this one. Maintain the low nutrient status of the soil. So if it's a lawn you're looking at, I always say you've got an eco lawn because it's ecological by nature and economic to manage because you don't have to buy any lawn improvers for it. Maintain existing drainage patterns. Generally speaking, they don't like very wet ground. There are one or two that will cope with very wet fields, but generally speaking, they prefer it to be well drained. So if you've got a good waxcap field, the chances are the drainage is about right to try and maintain that. Avoid disturbance or compaction. Disturbance, really by that I'm meaning ploughing, but we're not sure about compaction. When they looked on the continent where they pick a lot more fungi, and some research was done on the effect of picking wild fungi. And what they did was they looked at, I think it was probably chanterelle, and they had 3 sets of plots. One was completely picked. Every time a fungus put its head up, it was completely picked. One was just left without picking or anything. And the third one, they trampled up and down, up and down, up and down. And what they found was that the one that they trampled up and down on completely stopped producing fruiting bodies. And the one that they picked constantly just didn't make any difference, it just kept producing fruit bodies. So there does seem to be an issue with compaction. And this can be sometimes with these lovely old houses and their lawns. Very often the lawn is a sort of focus for wedding venues or, I don't know, the village fete or whatever. I'm not suggesting that the occasional village fete is going to have a big problem. But if you've got a lot of heavy vehicles and repeated use of a site by a lot of people, or a lot of vehicles putting marquees up or something, that could seriously have an impact on the fungi. Whether it's destroying the fruiting bodies or whether it's actually compacting the mycelium and causing a problem there, we're not sure yet. Avoid the use of fungicides. That sounds very obvious, but there we go. Moss, we don't know. We know that moss is very much a part of waxcap grasslands. There's no evidence to suggest that they are growing together in any kind of symbiotic relationship. And that actually is worth just saying a little bit more about. I've said that they get out-competed by the species of fungi that grow on the roots of trees in woodlands. What we thought was that the waxcap suite of fungi was actually recycling dead plant material. And it was assumed that it was probably quite deep litter that they were working on and recycling. But when somebody actually looked at it, what they found was they were expecting therefore to find quite old carbon. I think it's carbon 14 they were using, that they were looking at. And they were expecting to find carbon 14 that was 5-6 years old. And what they found was current year carbon. And that suggests that they are either parasitizing something and getting their nutrients directly from that, from that other organism, or that they are living symbiotically with another organism in a similar way that the tree fungi are living with their trees in the woodlands. The difficulty with that is that they cannot find what they would normally expect to find when that exchange takes place. There's that there are specific growth forms in the roots and around the outside of the roots where the exchange of nutrients takes place. And they can't find that with waxcaps. They just can't find it in the vegetation. They have found waxcap DNA in plant tissue, but it doesn't appear to be active. It's clearly something there that we still don't fully understand going on. All we do know is that all of the members of the suite living in waxcap grasslands are feeding in the same way, because that they can tell by using stable isotopes, I think. You'll tell that I'm not a chemist because I do struggle a bit with those, but the general idea is there. So there was no evidence of any interaction with our waxcaps and moss, so it's very likely that the waxcap suite of fungi and moss simply like the same set of edaphic characters in their habitat. And of course, what we're doing tonight, which is let's try and promote good management practice as far as we can in our waxcap grasslands, because they are overlooked and very important. That's probably pretty much enough from me. if you were interested, obviously, if you're wanting to get out there, there are various keys that you can access online. This is a particularly nice one, the Plant Life Waxcap app. If you Google Plant Life Waxcap, you'll come up, I think, with this link. And it's all about getting out there and recording wax caps and helping you to get familiar with the characters and how to put them together, and how you might decide there were different species on your site. I know that TWIC would be also very happy to hear about any records that you make. You can put them onto iRecord, and the British Mycological Society, their database of fungal records, it relates with iRecord. So there are lots of ways that you can record what you're finding. And when you do come across, you know, a little corner of grassland, or maybe even a big area of grassland that is, as far as you know, nobody's taking any notice of, it would certainly be worth bringing it to somebody's attention, either at TWIC, or if you go online, if you search for, what was it, Scottish fungi Google sites. That's an excellent site that will give you all the Scottish fungal recording groups. They're just groups of people that are interested in recording fungi and identifying them, and going out with them is a very good way of learning more about fungi and how to identify them. So I would certainly recommend you have a look at that website, which is Scottish Fungi, and it's the Google Sites one. Okay, I think. There we go. That's just a pretty picture to finish with. That's Hygroscybes splendidissima. This autumn on Hadrian's Wall, we did a waxcap surveying and workshop down there this year. Thank you for your time and for listening. I hope it's given you a bit of an idea, and I'll leave it to Natalie then to sort out any questions that there might be. I think I probably have to stop sharing, do I, Natalie?